Good morning, friends. Good morning, students. Today we'll discuss heme synthesis, right? So, first of all, the organ in which it takes place. There are two major organs. First is erythroid, precursor cells of bone marrow, right? And the second major organ is liver, right? Now, coming on to the site, which part of the cell where the synthesis take place. Remember that is supposed to happen in both the organs, that is mitochondria and cytosol, right? So you find it difficult to remember which part it happens. So I will give you mnemonic for that. Remember the mnemonic HUG, right? When you have to hug someone, you have to use two arms, right? So hug is for where you have two organs involved, right? So H for chemosynthesis and U for urea cycle, right? And G for gluconeogenesis, right? So these are the three pathways that are supposed to happen in both mitochondria and cytosol. First is our heme synthesis, which are we're going to discuss today. Second is urea cycle, right? And the third is gluconeogenesis, right? Now we'll start with heme synthesis proper. To remember it, first of all, remember that there are total 8 steps, right? And I already told that it's supposed to happen in both mitochondria and cytosol, right? So you can remember it that the first step is mitochondrial, right? And second, third, fourth, and fifth steps are cytosolic, right? And last six, seven, and eight steps are again mitochondrial, right? That's clear. The first step is supposed to happen in mitochondria. Second, third, fourth, they are cytosolic, and the last six, seven, eight are mitochondrial. Right now, uh, it is very difficult to remember the steps. So there is an mnemonic for that. You can remember it, and you can remember the steps. Right. So the mnemonic is: some great doctors palpate you undercover to produce perfect humans, right? So I will write down the steps later on to under this mnemonic to remember the steps. Now we'll start with steps proper, right? So the mnemonic is some great doctors, right? So S for succinyl CoA and G for great for glycine, right? So these are the two precursors from which heme synthesis will start. Glycine is the amino acid, and succinyl CoA comes from TCA cycle, right? So these two combine to form delta amino levulinic acid, right? And this step is first step, right? So it is mitochondrial, right? And this step requires vitamin B6, right? And the enzyme is ALA synthase, right? So B6 the vitamin that is required for the synthesis of delta amino levulinic acid, right? So if we have deficiency of vitamin B6 or pyridoxine, the heme synthesis will not happen and we will get anemia or sideroblastic anemia, right? Clear? Now some great doctors is clear: succinyl CoA, glycine, and finally delta amino levulinic acid, right? So the next step is some great doctors palpate. Palpate for bilinogen, right? And this is the second step, right? Second. And second step is supposed to happen in cytosol, right? The first step is mitochondria, second is cytosolic, right? And the enzyme is second. So you can remember by ALA dehydratase. So how to remember this enzyme? Second step is there di. Di for dehydratase. The enzyme is ALA dehydratase, and we will get phosphorylinogen, right? And this step is very important, right? Because it is inhibited by lead, right? So lead will inhibit this enzyme, and the reaction will not proceed further. And lead also inhibits the last step, second last step, that is ferrochelatase reaction. It also inhibits that reaction as well, right? Some great doctors palpate succinyl CoA from TCA glycine in the presence of ALA synthase will form delta amino levulinic acid, right? From ALA will synthesize phosphorylation by second enzyme D for di, and second ALA dehydratase, and will get phosphorylinogen. Now this phosphorylinogen will undergo deamination reaction by phosphorylinogen deaminase, right? And we will get uroporphinogen 1, right? So U for uroporphinogen 1, then it will form uroporphinogen 3, right? And the enzyme required is uroporphinogen synthase, right? And this uroporphinogen is converted into coproporphyrinogen, right? And in this step, deaminase, the amino groups are moved, right? And from uroporphyrinogen 3, we synthesize coproporphyrinogen, right? And this reaction is decarboxylation reaction, and the enzyme is decarboxylase. Now there is problem with uroporphyrinogen 1 in acute intermittent porphyria, right? And in this disease, the barbiturates are totally contraindicated, right? So why? Because these barbiturates are cytochrome inducers, right? So cytochrome inducers will increase the synthesis of heme as well, and will further elevate the symptoms of this disease. Now we got copropyrinogen. This will undergo copropyro oxidase reaction to form protoporphyrinogen 9, right? From protopyrinogen 9, we will get protoporphyrin 9, right? From here, we'll get protophyrin 9 to from we'll get finally heme by ferrochelatase reaction, and Fe2 positive is added, and we'll get finally the product that is heme, right? So just to revise well, Some great doctors palpate you under cover to produce protect humans, right? Some great succinyl CoA and glycine, right? To produce doctors for D delta amino levulinic acid by the enzyme ALA synthase, right? And then you undercover from delta amino levulinic acid will get porphobilinogen, and the second enzyme is ALA dehydratase from second from di. From PBG will get by deamination reaction the 4 amino acid groups will be removed to form uroporphyrinogen 1. From uroporphyrinogen will get uroporphyrinogen 3 by synthetase reaction, and from uroporphyrinogen 3 will get coproporphyrinogen by decarboxylation reaction. The carbon dioxide is removed. From coproporphyrinogen will get protoporphyrinogen 9, right, by oxidase reaction, write this. And from next P is for protoporphyrin 9, right? And from this will finally form humans, that is heme. And the reaction is ferrochelatase reaction is involved, and Rn Fe2 positive is added, and finally the proper heme is synthesized. So that finishes with our heme synthesis, and in the next video we'll discuss porphyrias in detail. And so please like the channel and subscribe the channel and keep on studying. Best wishes. Thank you.